Hello, dear viewers. Today, the topic that we are going to discuss is fungi, their general characters and classification. The lecture comprises of 3 parts. First, we will introduce the fungi. Second, we will discuss their general characters. And lastly, we will discuss the classification of fungi. So, starting from its introduction, fungi form a diverse group of spore-bearing heterotrophic living organisms. They are so distinctive from all other living organisms that they have been assigned to their own kingdom of fungi. At present, about 1 lakh species of fungi are scientifically known, and around 2 lakh more species are estimated to still remain undiscovered. Commonly, we know the fungi are causative agents for many plant diseases. For example, the plant pathogen that caused infamous Irish famine was the fungi, that is Phytophthora infestans. In humans, fungi are responsible for the ringworm of the body and athlete's foot disease. However, all fungi are not detrimental. The yeasts are of commercial importance in the production of bread, beer, wine. Other fungi are also used by pharmaceutical companies to produce drugs and antibiotics such as penicillin. Together with heterotrophic bacteria, fungi are the principal decomposers of the biosphere. Many fungi form important symbiotic relationships such as mycorrhizae, lichens. Now the second part, that's the general characters of fungi. We will start from the modes of nutrition in fungi. Like animals, all fungi show heterotrophic mode of nutrition. However, unlike animals, fungi are unable to engulf food materials. Therefore, fungi derive nutrients from the environment, from living, dying, or dead organisms. Based on this, fungi are divided into 3 categories. Number 1, biotrophs. Number 2, necrotrophs. And number 3, saprotrophs. Biotrophs. These obtain nutrients from living hosts without killing them. Many biotrophs secrete chemicals that make the plasma membrane permeable to sugars. As they leak from the host cell, the fungus absorbs them. While as majority of the biotrophic fungi remain confined to the intercellular spaces, a few are able to pierce through the cell wall by creating a hole and inserting a portion of the filamentous cell called as hostorium. The latter brings it close to the plasma membrane and makes it easier to absorb nutrients. Necrotrophs. These first attack and kill the living hosts and then absorb the released nutrients. Many necrotrophs secrete toxins that kill host cell. Fungal toxins damage the plasma membrane due to the loss of nutrient equilibrium that kills the host cell. Saprotrophs. These absorb nutrients from the dead organisms after they have died of other causes. Saprotrophs predominantly depend on the extracellular digestion, whereby digestive enzymes are secreted to break down the host polymers. Fungal body, about its cellular organization. All fungi are filamentous, except unicellular ones. The individual thread-like filaments are called as hyphae. A profuse branching network of hyphae is known as mycelium. The word mycelium is derived from Greek word myces, which means fungus. And also, because of this reason, the discipline that deals with the scientific study of fungi is known as mycology. The hyphae can be long and multinucleate cells known as cenocytes, for example, in water molds, or can be septate and cellular, for example, in sac fungi. A typical fungal cell is eukaryotic. A membrane-bound nucleus is present there, a nucleolus and membranous organelles in the cytoplasm. Mitochondria are generally abundant. Endoplasm reticulum is sparse and of smooth type. Ribosomes are of ATS type. Small vacuoles are present. Dictyosomes are rare, and plastids are totally absent. Like roots, hyphae grow only at the tip. The cell wall of all fungi except Oomycota, particularly the innermost wall layer, is rich in chitin, a polymer of N-acetyl glucosamine arranged in microfibrils just like in cellulose. The chitin layer is followed by a predominantly proteinaceous layer. External to this layer and outermost to it is a thick layer of polymerized sugars of complex structure. Another striking feature of hyphae is the nature of cross walls called as septa. Lower fungi generally lack septa. For example, hyphae in Myxomycota are coinocytic with no septa. Hyphae in Oomycota and Zygomycota are usually non-septate. On the other hand, hyphae in higher fungi like Ascomycota and Basidiomycota are septate. In Ascomycota, the septum has a simple pore. In Basidiomycota, pore has a complex structure with a collar or hemispherical cap on each side. Now, one more important feature in fungi is fruiting body. In case of Ascomycota and Basidiomycota, some of the mycelia, they form a large, compact and highly organized structure called as fruiting body. The fruiting body is the chief means of spore production. We have three types of hyphae. Number one, generative hyphae, which are thin-walled and produce spores. Skeletal hyphae, which are thick-walled and unbranched, and binding hyphae, which are thick-walled but they show profuse branching. These adhere to other types and bind them into a solid structure. Now about spores. The spore formation is a characteristic feature of fungi. The spores are resistant resting stages, which are the primary means of reproduction, dispersal, and survival. These are produced either asexually or sexually. Asexual spores. In the Zygomycota, asexual spores produced are called sporangiospores. The sporangiospores are produced inside the large swollen tip of a hypha. At the tip of the hypha, cytoplasm and many nuclei accumulate. A septum seals the region off from the rest of the hypha, and each nucleus organizes cytoplasm around it to form a spore. When the hyphal wall breaks down, the spores are released. On germination, the sporangiospores are non-motile, except in lower fungi like Myxomycota and Oomycota. In the Ascomycota, Basidiomycota and Deuteriomycota, asexual spores are formed in conidia. In other words, spores do not form inside a sporangium. The simplest case of conidia formation is where the tip of a hypha forms septa, cutting off several uninucleate cells, each forming an individual spore. On germination, conidia are always non-motile and grow out as a hypha that develops into a new mycelium. In fungi, the spores are small in size and light in weight. They are able to get dispersed easily to great distances. Many fungi have long-lived spores that can withstand environmental fluctuations. The most common type of resistant spore is a chlamydospore, especially in soil fungi. Chlamydospores are formed when a mass of protoplasm rich in reserves of oil or glycogen accumulates within a short length of hypha with thick-knit walls. As the conditions become unfavorable, the rest of the hypha dies. Only the chlamydospore survives until the return of favorable conditions. Another type of resistant structures that develop generally in biotrophic fungi is called as the sclerotia. The sclerotia germinates depending upon the conditions favorable to the host. One more important phenomenon that is in fungi is called as heterokaryosis. Almost in majority of the fungi, sexual reproduction does not involve the production of distinct gametes. Instead, hyphae of one mycelium fuse with that of another. These two mating hyphae are morphologically similar but physiologically different and are designated as plus and minus. The fusion of two hyphae called plasmogamy is not immediately followed by the nuclear fusion called as karyogamy. The fused hyphae that contain two types of nuclei, plus and minus, forms a dikaryon. It grows for some time, a condition called as heterokaryosis. Ultimately, the karyogamy occurs wherein the two nuclei of compatible mating types fuse to form a diploid nucleus. This is immediately followed by meiosis, during which synapses and crossing over takes place. As a result of this, plus and minus types of spores are produced that have recombination of alleles. The spores that number millions blow away and each germinates into a new plus or minus mycelia. Parasexuality. In some fungi, the compatible nuclei fuse prematurely to form a diploid nucleus, followed by meiosis, a process termed as parasexual cycle. Then the hypha returns to a heterokaryotic phase, but with a haploid nuclei. When these nuclei participate in the formation of conidia or sporangiospores, the spores in that case have different genotype than the original parental nuclei. In the deuteriomycota, the parasexual cycle is the only means of genetic recombination. Now the third part of the lecture, that is the classification of fungi. In the traditional two-kingdom system of classification, fungi along with bacteria were grouped together under plants because they produce spores and have cell walls. However, more recently it has become now clear that the living organisms grouped together as fungi are clearly different from plants because they lack plastids, their walls are generally devoid of cellulose, the main body is filamentous rather than parenchymatous. They differ in many biochemical pathways. Presently, the classification of fungi is in a state of flux. It is continuously changing. More recently, with molecular evidences available, the classification of fungi has witnessed a continuous splitting and regrouping of different divisions under the kingdom fungi. The lower fungi like slime molds and water molds, traditionally grouped together under fungi, are now excluded from the true fungi and are included under the kingdom Protista. Still, for the purpose of simplicity, all the organisms that were previously known as fungi, including the true fungi, can be broadly recognized under the following 7 divisions, or divisions are also called as phyla. And the kingdom fungi, these 7 divisions are Myxomycota, Oomycota. They are not nowadays considered to be the true fungi. Then the true fungi, Chytridomycota, Zygomycota, Ascomycota, Basidiomycota, and the last Deuteriomycota. We will now discuss the diagnostic characters of each of these divisions one by one, starting with Myxomycota. Myxomycota are commonly known as slime molds. These are quite distinct from the true fungi. These are heterotrophic and form spores, but they lack cell walls and show a unique body organization. The body is a large mass of protoplasm containing thousands or millions of nuclei, all in the same cytoplasm and covered by only plasma membrane. This mass of protoplasm, called as a plasmodium, is capable of migrating over a substrate, just like amoeba. The second division is Oomycota, commonly known as water molds. These are aquatic for part of their life cycle. Slime water molds, they live in water, soil, and even few are parasites of plants and animals. They range from unicellular to highly branched filamentous forms. The filamentous forms somewhat resemble the hyphae that are characteristic of true fungi. For this reason, they were grouped under the fungi until very recently. Unlike true fungi, their cell wall is composed of not chitin but cellulose. These reproduce both sexually and asexually. The asexual reproduction is by means of not non-motile but motile zoospores. The sexual reproduction is of oogamous type. Now the third division is Chytridomycota, commonly known as chytrids. These are predominantly aquatic fungi. They occur also in soils from ditches, ponds, and streams. They live as saprophytes on substrates such as dead insects and a few are plant pathogens. They are either unicellular throughout their life cycle or form a small non-septate mycelium and thus coinocytic. Like other fungi, their cell wall contains chitin and store glycogen. They are distinguished from other fungi primarily by their motile zoospores and gametes. The next division is Zygomycota, commonly known as conjugation fungi. The phylum is named for its characteristic zygospores, which develop within the thick-walled structures called zygosporangia that remain dormant for longer periods. The black bed mold, scientifically known as Rhizopus stolonifer, is the most familiar example of Zygomycota. These are mostly terrestrial. They live on decaying plant and animal matter in soil occurring mostly as saprophytes. The next division is ascomycota. These are commonly known as sac fungi. They show filamentous growth forms except the unicellular yeasts. Characteristic feature of these fungi is the ascus, a sac-like structure containing haploid sexual spores termed as ascospores. Unlike zygomycota, which produce spores internally within a sporangium, the ascomycota produce the spores externally as conidia. The next division is basidiomycota, commonly known as club fungi. These include mushrooms, puffballs, and important plant pathogens, the rusts and smuts. The spores produced by these fungi following the meiosis are called basidiospores and are borne on a club-shaped structure called basidium. The next division is called as deuteriomycota, commonly known as fungi imperfecti, often called as fungi imperfecti because sexually reproducing stages are absent here. These are an artificial assemblage of fungi for which only the asexually reproducing state is known. By this, we completed the classification of fungi. Hope you have now a better understanding of fungi. Thank you.